The overall goal of this procedure is to analyze intact proteins larger than 100 kilodaltons in a sensitive manner by MALDI-MS spectrometry. This is accomplished by first preparing the matrix solutions. The second step is to deposit the thin layer solution on the target. Next, the sample and the mix of matrices are deposited on the target. The final step is to observe the sample spots under the microscope. Ultimately, a MALDI-TOF mass spectrometer is used to analyze intact proteins and record mass spectra.
The main advantage of MALDI mass spectrometry over other analytical approaches, such as electrospray ionization, is that it is more tolerant to salts, contaminants, and detergents.
Another advantage of MALDI is that it generates ions carrying fewer charges compared to electrospray, therefore acquiring and interpreting MALDI data is quite straightforward.
To begin the recrystallization, pour 10 milliliters of 40% ethanol in a Pyrex flask, then add 600 milligrams of a matrix. Using a water bath and a resistance heater, warm the matrix solution and stir it until the matrix is completely dissolved. Repeat the previous steps until the solution is saturated. Allow the solution to cool slowly at room temperature for several hours and then store at 4 degrees Celsius overnight. If crystals are not formed, scratch the inside of the beaker just below the solution surface using a glass rod to induce crystallization. Finally, collect the matrix crystals by filtration. Rinse the residual crystals in the flask with a minimum amount of ice-cold solvent and filtrate them. Once filtration is complete,